Welcome, everyone. My name is Mutiat Osman. Today, I'll be talking about my research on DGGM. DGGM means a dynamic Gaussian graphical models, and I'll be using DGGM subsequently. And we want to be using this framework to detect persistency and connectivity in organoid network under a time-varying covariance. This work is supervised by Dr. Kim and co-advised by Dr. Yin. Now, let's talk about the introduction. Now, the very first thing we are all going to agree with me is that we have one thing in common regardless of who we are, is the fact that we are human and we have a working brain. But then has anyone ever thought about what really happens in the brain when we have diseases of the brain or distortions of the brain? Well, this is the focus of my work. Research has shown that the human brain has approximately 6 billions of networks, and they are responsible for memory, movement, reasoning, personality. And when this connection, it requires a stable dynamics network, which will adapt over time. And when there's any small disruption in this network, it can lead to profound functional changes, which is what I talked about earlier, or neurodegenerative disease like the Alzheimer and Parkinson, which are the two most common neurodegenerative diseases. Now, let's look at the existing method in brain research and how we have gone so far over decades of research. Now, we have a post-mortem approach. Post-mortem approach requires someone to die, and then they can do a research, but we have a major limitation in terms of consent and some other factors. Also, animal model has been a major source of testing, but research has shown that for the past decades of research on animal testing, it has not gotten us close to understanding the complexity of the human brain itself. And hence, there's been a lot of news as regards reliance on animal testing, and they're trying to move away from that. We had a major breakthrough on the 2D cell culture, which is the two-dimensional cell culturing, which requires a flat monolayer, and also because of the complexity and the 3D nature of the human brain, it's been a major stepping stone, but it does not properly address the complexity of the human brain. Now, there has been several brain imaging techniques like the functional MRI, the PET scan, which all these approaches are non-invasive. Non-invasive means that you do not penetrate the brain. So they put some microelectrodes on top of the skull, and then they take some of the brain activity measure. Like for functional MRI, you might have to engage in some form of exercise, maybe reading or some form of activities, and then they take the brain recording activities. Well, with all of this limitation, it's been a problem with research on how we can further explore understanding the complexity of the human brain. And we have the brain organoids. Think of brain organoids as the human versions of, as growing the brain outside the human skull is the lab-grown version of the human brain. That is, for the first time, they can actually bring the human brain outside the human skull. And this time around in a lab setting. That's why we call it in vitro. And because brain organoids can reorganize themselves, there are many miniature versions of the human brain that they can reorganize themselves. And this has been a way of studying the human brain real time on how these brains are forming their network and how they are reorganizing. And also to support this work, to support the importance of this work, NIH had a news release that they had their first dedicated organoid center to reduce the reliance on animal testing. All right, let's move on. Let's talk about the main analysis and gap in knowledge. I've done a rigorous literature research on this work, and we're going to be looking at 3 main critical gaps and the team. Now, when I say Gaussian graphical models, that's the GGM, which I'll be using. So static GGM, they have not properly accounted for temporal variation, which has limits the insight in understanding dynamic network reorganization. This work was proposed by Monti et al. And the second thing is understanding the hub detection itself. When I say hub means one of the most important nodes or in the brain. So like we said the human brain, they naturally form a network in the brain. And we're trying to understand some of the most influential nodes, like the node that is very important in the brain. And when something happens to this kind of cell in the brain, then we have problems like in terms of communication or movement. Also, there has been a data pipeline mismatch, like high-dimensional microelectrode array pipeline, which will be adapting the CITETOL 2024 approach, MINAP, where they are able to understand the static approach, but yet nobody has properly addressed the dynamic graphical modeling framework. All right, let's talk about the gap. We have the gap currently is that we do not have a unified framework that exists for detecting the time-varying functional ops in organoid network, which is what our work is proposing. Now, let's talk about the case of dynamic hop detection. Brain organoids are validated models of human neural development, yet analytical to consistent lag behind record activity. There has been static GGMs. Dynamic GGMs represent the necessary methodology advance, which can extend the graphical modeling into a time-varying settings. And within a dynamic framework, we can actually understand how these functional networks are emerging and reorganizing in development organoid circuit over time. So we're not just doing at a single time point. We're looking for something that can address dynamic time-varying functional connection in the brain. Now, the problem and hypothesis that we're able to formulate from this rigorous literature review that we've done is that how can we functionally develop a hop nodes image, and how do they evolve in organoid neural network across different developmental time? And our central hypothesis is organoid neural network, they naturally exhibit a time-varying hop structure that can reliably identify and characterize using our DGGM. The main objective is for us to develop and validate a DGGM-based hop detection framework, which we are going to be applying to high-dimensional microelectrode array recordings of brain record by leveraging on the CITETOL paper of preprocessing our pipeline. So here are my references. This work is in collaboration with the bioengineering department here in ANC, which is why Dr. Yun is co-advising this research with Dr. Kim. And also, we are building a methodological framework. So we're not just, as far as the data generation process is going on in bioengineering, we are building a framework that can properly address the kind of data the organoids network is generating, which is because they are very high dimensional and also because they are heterogeneous in nature. So there's a need for a new framework that can properly address this. Thank you so much.